Hi everyone, now we are going to discuss about what is light microscope, the phase contrast and dark field microscope. Myself Srishma PV, second MSc Biology, St. Mary's College, Thrissur. Mainly we are going to discussing about the light microscope and phase contrast microscope and dark field microscope. First of all, we can discuss about what is light microscope. In 1519, F.S. Janssen and Zacharias Janssen constructed the first simple light microscope. Then more details is not required. The light microscope is an ideal and essential instrument for a biologist, and the simplest form of light microscope consists of a single glass lens in combination, and all modern light microscopes are made up of more than one glass lens in combination. Then the picture is here. The light microscope that consists of an eyepiece, and it have an arm and no piece, an objective lens and stage, diaphragm and coarse adjustment and fine adjustment and light source and base. This is the overall picture of a light microscope. Then we want to discuss about what is the principle behind the light microscope. The light microscope operates on the principle that light energy will passes through and around 18 objects such as microorganism, and with the aid of lens form a magnified impression on the visual sensory layer of the eye. Then the main components of the compound light microscope include a light source that is focused at the specimen by a condenser lens. The source of light is usually the sun or the ambient indoor light. Then how does the light microscopy work? The light microscope are compound microscope, and which means they contains at least two lenses. The lenses are curved piece of glasses or plastic that bend rays of light and can magnify objects, that making them appear bigger than they actually are. The light microscope show here magnifies an object in two stages. Light from the mirror is reflected up to the specimens or the object to be viewed into the powerful objective lens, which produce the first magnification. The image produced by the objective lens is then magnified again by the eyepiece lens, which acts as a simple magnifying glass. The magnified image can be seen by looking into the eyepiece lens. Then the important factors in light microscopy is the magnification and the resolution and the contrast. Then mainly four types of light microscope are the bright field microscopy and dark field microscopy and phase contrast microscopy and fluorescence microscopy. Then we want to go we Then we want to discuss about the phase contrast microscopy in detail. The phase contrast microscopy, first described in 1934 a Dutch physicist Fritz Zernike, and the contrast enhancing optical technique that can be utilized to produce high contrast images of transparent specimens such as living cells, microorganisms, thin tissue slices, fibers, glass fragments, and subcellular particles. The phase contrast microscopy is a special adaptation of the light microscopy and helps to obtain a clear picture of living and unstained cells. The adapters convert minute difference in phase changes in transmitter light due to the refractive indices of all cell organelles into perceptible shades of grey. This allows organelles of the living cell to become visible with fair contrast in them. The picture of phase contrast microscope is here. Then mainly it have an angular ring and a phase ring and a SPL property. The angular ring is between the condenser and light source. Then the phase ring is between the objective lens and the image plane. The SPL property, both the rings allows partial light to pass through it, and the rest is blocked. The pathway of light source through the phase contrast microscope is the flowchart is here. The movement of light, the light from the light source, the light is passes through the annular ring, then the condenser, then the specimen plate and the objective lens, then the phase ring and the image plane. The image is detected from the image plane. Then The working principle behind the phase contrast microscope is the regions of different composition likely to have different refractive indices. Normally, such differences cannot be detected by our eyes. However, the PCM, there is an interference of light. There is an interaction of two light waves which lead to the formation of resultant waves, the constructive interference and the destructive interference. The picture of the constructive and the destructive interferences are the destructive is that, and the constructive is here. Then the light passes through the condenser via the angular ring. After reaching the specimen plate, the two types of beams are formed. If there is no specimen in lens, the surrounding wave that is represented by the S and the particle wave that is represented by the letter P, and the P is equal to S if there is no specimen in lens and there is no interference. If the lens contains the sample, the light beams gets diffracted because of different density at different regions of sample. Then the surrounding wave and the diffractive wave become P is equal to S plus D. Either the constructive interference or the destructive interference may occur. Then the ring-shaped illuminating light that passes the condenser annulus is focused on the specimen by the condenser, and some of the illuminating light is scattered by the specimen and the remaining light is unaffected by the specimen and forms the background light. In a phase contrast microscope, the image is improved by two different methods, the negative phase contrast and the positive phase contrast. When the in negative phase contrast produces destructive interferences. Thus, the image obtained is like inner region of the sample is bright and the outer region of the sample is darker and the surrounding lens is opaque, and the positive phase contrast produces constructive interferences. From it, the image of the specimen obtained is the inner region of the sample is darker and the outer region of the sample is brighter. Then the surrounding lens is also opaque. Then we want to discuss about what is the dark field microscopy. The picture of dark field microscopy is here. Then the dark field microscopy is used to examine the live microorganism that either invisible in the ordinary light microscope, cannot be stained by standard methods, or are so distorted by staining that their characteristic then cannot be identified. Instead of the normal condenser, a dark field microscopy uses a dark field condenser that contain a opaque disc. The disc blocks light that would enter the lens directly. Only the light is reflected off the specimen enters the objective lens. Because there is no background light, the specimen appears light against black background, the dark field. Then the major principle of the dark field microscopy is the dark ground microscope creates a contrast between the object and the surrounding field such that the background is dark and the object is bright. The object and ocular lens used in the dark ground. The microscope are the same as in the ordinary light microscope. However, a special condenser is used which prevent the transmitted light from directly illuminating the specimen. Only the oblique scattered light reaches the specimen and passes on to the lens system, causing the objective to appear bright against the dark background. Then we want to discuss about the major advantages of dark field microscope. A dark field microscope is ideal for viewing objects that are unstained, transparent and absorb little or no light. These specimens often have similar refractive indices as their surroundings, making them hard to distinguish with other illumination techniques. You can use dark field to study marine organisms such as algae and plankton diatoms, and insects fibers, use yeast and protozoa, as well as some minerals and crystals in polymer and some ceramics. You can also use the dark field in the research of live bacterium as well as mounted cells and tissues. It is more useful in examining the external details such as outlines of grains and boundaries and surface defects than internal structures also. Then the major applications of the dark field microscopy is used for diagnostic of syphilis and viewing blood cells, and also for viewing the bacteria and viewing different types of algae and viewing hairline, metal fracture and viewing diamond and other precious stones and viewing shrimp or other invertebrates. From this picture we can see the two bright field and the dark field structures of cells. The two differences are given in these pictures. Then the major disadvantages of dark field microscopy: the dark field image are prone to degrade description and inaccuracies. The specimen that is not thin enough or its density differs across the slide may appear to have artifacts throughout the image. And the preparation and quality of the client can grossly affected the contrast and accuracy of a dark field image. You need to take special care, that is this line stage known as light source for free from small particles such as dust, as the will appear as a part of the image. And similarly, if you need to use oil on water on the condenser and slide, it is almost impossible to avoid all air bubbles. Then thank you for watching this video.